Hello, everyone. I'm Christy Risk, Senior Editor at GenomeWeb, and I'll be your moderator for today. The title of today's webinar is Development of a Wastewater Analysis Protocol for SARS-CoV-2 using Quantitative Real-Time PCR, and our sponsor is Agilent Technologies. Our panelists today are Dr. Augustin Pieri, Technical Director at WEC Laboratories, and Dr. Sharon Israeli, Product Manager of PCR and qPCR reagents at Agilent. You may type in your question at any time during the webinar. You can do this to the Q&A panel, which appears on the right side of the webinar screen. And if you look to the tray at the bottom of your window, you'll see a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Dr. Pieri. Please go ahead.
Thank you for the introduction, Christy, and good morning, everyone. So today I'm pretty excited to talk about this method that we've developed for the determination of the genetic signal of SARS-CoV-2 in wastewater. I want to give a little bit of background on our lab first. So we're pretty much a full-service environmental laboratory, and we do things like drinking water and wastewater. Most of the work we do focuses on regulated contaminants. These are things that have limits in place and established methods already. A big chunk of our business actually comes from the determination of emerging contaminants in things like wastewater and drinking water. These kind of range from pharmaceuticals in drinking water to organometallic species in wastewater and kind of everything in between. And that includes also viruses. So we're located in beautiful Southern California here, which is interesting because we actually see a huge amount of, or we're seeing a huge amount of cases for COVID-19. So if you look at a heat map of the United States, we kind of are at the top of the list in terms of clinical cases. And being here in Southern California, Los Angeles County, we're actually seeing it firsthand and seeing how of a big effect it has on society. So to give a little bit of background on this virus that I'm sure everybody is very familiar with at this point. It's a SARS-type virus similar to some of the other pandemics we've had recently. And what's interesting about this is that it's the virus is shed conventionally through the respiratory tract, but it's also shed through the gastrointestinal system, which gives us kind of a second way of monitoring this, which is a little bit useful, especially for this method that we're talking about today. So as everybody in the whole world knows, I think this is becoming a pretty serious problem. It's a global pandemic, and this virus is causing a huge amount of stress on the healthcare systems in a lot of countries. So this is people showing up in hospitals sick or showing up in emergency rooms sick and putting in extreme stress on these systems. In addition to the stress on the healthcare providers, there's also a significant economic effect in society. So a big issue that we're seeing in a lot of places is that as things are closing and mediation, kind of approaches are taking place, and closing businesses and closing schools and putting people in quarantine, there's a significant economic risk here that we're kind of experiencing firsthand. So because of these issues, a lot of local and state and federal governments are putting in steps to minimize the transmission and growth of this virus and see if we can stem it. And these are things like putting in mask protocols or forced quarantines or closing down businesses or entire groups of businesses while this is kind of under control. And a lot of the work that people are doing to kind of mitigate these effects are actually using individual testing and contact tracing, both of which are a little bit cumbersome. All right, so if you have an example here, this is a population of 9 people. If you have, say, 3 of them who are positive COVID-19 cases and symptomatic, you can get those people tested and do individual tests on them, and then look at who they came in contact with, do a little contact tracing, and test those folks as well, and then kind of isolate it. And you have a pretty good way of minimizing the transmission of this virus. And it works pretty well, for a small population of like 9 people. What's interesting about this virus, though, is that it turns out there's a significant percentage of infected people who are asymptomatic. So let's say that those 2 people on the right are actually asymptomatic. So if they don't have symptoms, they're probably not getting tested. So we're not doing contact tracing. So those people are just going to be floating around the population, potentially getting other people sick. So if we're not able to test them, they'll get their neighbor sick, which in turn will get the rest of the population sick, which is kind of easy to manage if it's a population of 9. But what happens if it's a little bit bigger, or what if it's significantly bigger? Then it becomes pretty challenging to try to test everybody and who they come in contact with and get an idea of what's going on there. So what we would like, a better approach, I think, to add more information. It's not going to be a replacement for individual testing and contact tracing, of course, but it'd be another tool to have the decision makers be able to make more informed decisions. Would be if we could aggregate all of these people into a sample, say all of the asymptomatic people into one sample, and then mix those with all of the people who are symptomatic, and kind of mix them together and get an idea of what the population actually looks like. That'd be great, because then you can have kind of a composite sample and get a big picture overview with as much resolution as you would like to figure out what percentage of the population is sick, or if infection is trending up, trending down. And this is kind of where wastewater-based epidemiology is useful. So this is something that really hasn't been, it's not new. This is something that people have been using for a long time, used it for a lot of different viruses, for even things like drugs of abuse and other kinds of things in wastewater streams. But it's kind of interesting because you can use wastewater as a community aggregate of everything that's going on. So you can get, I think it's load. So you can, for example, if you had the society of homes and neighborhoods and apartments and schools and dorms and hospitals, all feeding into a wastewater treatment plant, and if everybody's not sick, then the wastewater comes out non-contaminated or without the genetic signal for SARS-CoV-2. But let's say you have some of these folks sick, so a couple houses in your neighborhoods, maybe some apartments, a dorm or two at a university, and of course the hospital where a lot of sick folks are going. Then all of these individual sites are feeding the wastewater treatment plant here on the bottom right, and that will aggregate together. So what's nice about this wastewater-based epidemiology is that if you choose your sample points well, you can kind of get an idea with a high degree of resolution of what's going on. So if you say pull sample points here at the points, or pull samples at these points where these homes or apartments or dorms or whatever it is meet the wastewater treatment plant system, then you can kind of see what's upstream of that. So if you're pulling at that first circle on the left, you can say, oh, that these individual homes have no infections, but the communities have some percentage. Well, the apartment has maybe a little bit more, and then the dorm has more. So you can kind of get that resolution. Or if you want to look at big picture, you can just pull a sample right before the treatment plant, and that'll aggregate everything that you're looking at that's feeding that system. The thing to remember here is that it's a wastewater stream is pretty tricky. It's pretty complicated. There are a lot of factors to consider. So if you're trying to get an idea of what this means in terms of who's sick or how many people are there, you'd have to take into account factors like what is your flow composed of? Is it raining? Is there stormwater runoff? Is there agricultural runoff? All those kind of things would kind of take into place. So it's not so straightforward, but it makes it a nice tool to, I guess, see what's going on upstream of those. So some use cases of this kind of approach where it would be pretty useful or helpful would be kind of small catchments of very tightly controlled, I guess, living environment. So it'd be things like dorms at schools or boarding schools, or things where people all live and work and do things in the same place, or like assisted living communities where you're going to be able to say, okay, all of the discharge from this site is due to the people that live and work here only. So it's easy to kind of get an idea of what's going on. It's also useful for monitoring what happens during these mitigation steps. So let's say if you are a school, a university, and you want to open your university again, you can start looking at the wastewater coming out of the dorms and get an idea of what's trending. Is the infection rate trending up, trending down? What's going on? You can kind of gauge what's about to happen from looking at the wastewater. You can also do the same thing with opening and closing of facilities and businesses in the city. Say you have a community where you close barbershops and bars. You can see if that had an effect on bringing the infection rate down. And conversely, when you open them up, you can see if those infection rates are going back up. Now, one thing that we've had a few questions now, is kind of the limitations of this. And it's important as you do this testing and get this data to understand what it does and doesn't tell you. So even though we can report a number, right, it's the genetic copies per liter of wastewater, it's very, very difficult to equate that number to the number of infected individuals upstream of that sample site. And like I mentioned earlier, you have to take a ton of things into account, things like the flow in your wastewater system. If it's raining, how many people are there? Is the person who's shedding virus early in the infection, late in the infection, are they shedding a lot or a little? Or are they maybe not shedding any? It's, there's a lot of factors to be considered. So it's kind of tough to say, oh, this concentration of SARS-CoV-2 equals this number of people sick. So the best way to use this data, I think, is as a trending indicator, right? So you can see if it's going up or down. And as kind of another ancillary benefit, or not, a significant benefit of using this is the speed of which we can get data. So if you take an example of comparing individual testing to wastewater testing, right? So if you have a person get sick at day 0, they're going to start shedding the virus through the gastrointestinal tract maybe around day 2. And by the time it gets to the wastewater treatment plant in day 3, you can have a sampler already going, collecting a sample. And again, these samples should be collected in a composite fashion, right? So you're doing a 24-hour composite to get a representative idea of what you're seeing in the stream. Pull the sample on day 4, send it to the lab, maybe have to ship it over a distance, get the results back from the lab, or get the sample to the lab, and maybe they get results in 2 days. So by day 6, they have data. And by 7, the person who's collecting the sample has some sort of actionable information where they can make a decision or help inform a decision based on all the other data points that they have in terms of individual testing and all that stuff to see what should be happening in this situation. And if you compare that to individual testing, an infected person, if they're even showing symptoms, remember that some of these are asymptomatic, might not be showing symptoms until day 8. At that point, they'll go to a testing site, get tested. It takes a day. It takes another day to get the result. And by the time that you have that positive result on day 12, then you can have actionable information from that side, which is kind of it's almost a weak difference between what kind of information you get from wastewater testing to individual testing. So you can use this as kind of a leading indicator of infection rates. And sure enough, a lot of data actually does support that, right? So this kind of wastewater monitoring is shown in a lot of studies and literature in Europe and other places that it does show it's a leading indicator to clinical cases. So you can get 4 to 5 to maybe even up to 8 or 9 days lead time of a rise in infection rates when you compare the clinical cases to what you see in the wastewater. So this is actually really useful to be able to tell if something is about to happen. So if you know that an infection outbreak is going to happen, you can maybe take steps to mitigate it before a bunch of people show up sick in a hospital, which is very useful, especially if you can get this data out quickly. But like I mentioned before, it's really important to look at this as just a trending value, right? It's not going to be something useful if you say, oh, this means I have 22 infected people in this community of 200. It really only says, oh, this trend is increasing or the infection rate is decreasing, so you can kind of get an idea of what's going on in your population as a whole and not really pull out individuals. So when people are pulling these samples, it's kind of important to formulate a sampling plan that makes sense, right? Like if you're going to be doing if you want to look at very specific high resolution data, you want to be pulling samples right before or right after a community or a dorm or a university or something, so you can see what that particular site is looking like. And one thing you can also do is look at community events. So like say, if you want to measure how well a quarantine shutdown worked, you'd have to have some sort of baseline before. So you want to measure a few days or weeks beforehand to get an idea of what the baseline infectivity rate is. And after you put in your change, either opening or closing businesses or schools or whatever, then you can see if that rate changes or that infection rate goes up or down. And then one thing that I also want to stress is that it's important to take a good quality sample. So this is a picture here on the right of an ISCO sampler, which is a 24-hour compositing device where you can pull samples, say, over 24 hours and get kind of an idea of what actually is going through your treatment plant over the course of a day. So if you just grab one grab sample in the middle of the night when it's low flow into the treatment plant and nobody's contributing to that stream, you're going to get maybe a low bias if you see anything. But a 24-hour composite would give you a better idea of what actually is going on in your sample. So it's important to take good quality samples using these compositing devices. So kind of want to talk about the protocol that we're doing here in our lab. So like I mentioned, a good quality sample is important to begin with. So if you're doing this protocol, first you want to go down and collect a sample. So you go down to your local wastewater treatment plant. Let's see if I can get this guy to show up. Yeah, so you head down to your local wastewater treatment plant and deploy your ISCO sampler and collect a 24-hour composite at either the plant influent or a particular sample point somewhere along the treatment line. And in this approach, we're using a 50 ml sample. So you collect your sample in sterile clean tubes. Samples look like this. Now, wastewater is pretty tricky, pretty complicated. We get the whole range of cleanliness. So this is a particularly clean sample, but they get much, much, much dirtier than that. So once the sample comes into our lab, we pasteurize it at 75 degrees for 90 minutes. And the reason we do this is for a couple of reasons. So one, we want to be able to inactivate any virus that's potentially in there to keep folks safe. We don't want to have a potentially infectious sample floating around the lab here, even though a lot of the literature suggests that maybe there's very little risk of infection from the SARS-CoV-2 in a wastewater sample, because it does have to travel kind of a long ways to get into the wastewater stream. So it potentially is not so infectious. But also something that needs to be taken into account is that wastewater just has a lot of nasties, right? There's a lot of stuff in wastewater, a lot of bacteria, other viruses, other things that we really don't want to have in the sample. So we use pasteurization to kind of kill everything and be able to have a reasonably clean-ish sample to work with. But besides, the cellular debris and the bacteria and all the other stuff that's in wastewater, there's a lot of total suspended solid. The TSS is very high in most wastewater influents. So to eliminate this solid material and the suspended stuff, we filter the samples through a 0.22 micron filter. So we filter that, filters pretty quickly depending on the dirtiness of the sample to eliminate a lot of the solids. And from there, we have a reasonably clean sample free of suspended solids. Now, because it's wastewater, and a lot of these wastewaters have some industrial discharge components to them, the samples are still kind of dirty. There's a lot of potentially inhibitory things in there, organics, metals, all kinds of stuff in there. And also, the sample's pretty dilute. At this point, we still have 50 mils of a somewhat dilute concentration of the SARS-CoV-2 RNA, so we do a concentration from here. And we actually concentrate that on a molecular weight cutoff filter, a 10,000 Dalton filter that we spin down, and we put the whole 50 ml volume through one of these little filters and bring it down to a couple hundred microliters, about 400 microliters. And from there, now we have a reasonably concentrated sample, but it still has a lot of that junk in it, right? It still has a lot of the organics, a lot of the metals, and all the things that are potentially inhibitory to the PCR experiment. So from there, we'd have to do an extraction to clean up the sample, get the RNA out, and stabilize it, get it ready for analysis. So for this, we use a spin filter system. We use the absolutely total RNA extraction kit from Agilent, and we do a bunch of washes on this to make sure we can remove all of the bad stuff, the stuff that would interfere with the PCR reaction, and we're left with a extract of about 100 microliters. So this will do actually in duplicate. So we're looking at both the N1 and N2 probe primer sets as recommended by the CDC. And we use a Brilliant 3 master mix from Agilent also, and then do this kind of in two separate runs to be able to get an idea of what's going on in each probe set. So one thing that's important to consider when doing this stuff, and something that we thought a lot about, especially developing it and kind of implementing the protocols in environmental samples that are very regulated and require a lot of quality control considerations. So we wanted to make sure that we had a system that was very robust and we had a high degree of confidence in the result. So to give you an idea of what a plate would look like, for example, if you had a fully loaded batch with all the QCs, we first have our calibration curve. So we're doing a calibration curve of 5 orders of magnitude, 5 different wells. We run these in triplicate, just to be sure. And then we have our batch QCs that come in after that. So there's going to be no template control. So we just look at the background of the reagents, make sure nothing's contaminated. We have a negative control, which is OC43. It's another human coronavirus that will not trigger a response in the N1 and N2 probe primer sets. And we also do a second source positive control. And the reason we do this is to confirm that the calibration curve is good, that the calibration curve is accurate, that the standards we're using for that agree with a different source from a different vendor. So we have these two to use as an independent calibration verification standard. And one thing that I think is important for this method is doing matrix spikes. So we include a matrix spike with every batch, and this is going to be a process control that gets carried through the whole extraction procedure. So we inoculate the incoming sample with SARS-CoV-2 and inactive SARS-CoV-2, but it goes through the whole procedure of pasteurization, filtration, concentration, extraction, everything. So we can really assess how the whole method is behaving, and if there's any weird stuff, anything problematic or potentially problematic, it would show up in the matrix spike. Let's say if we had an issue where maybe the filters were taking the target out, we would see zero recovery, very low recovery from the matrix spike. So it's important to have this as one of the key quality control requirements. And from here we have a space for 23 samples, so we'll run these in duplicate. And one thing that we also do, because wastewater is tricky and complicated and has a lot of different things going on with it, we also do an inhibition control on all of these. So we'll run an extra well of the sample that has the same spike as that second source positive control in it. So we can kind of positive spike the sample in the PCR plate and get an idea of if there's any inhibition. So if we see a difference in CQ value between the second source positive control and the inhibition control for a particular sample, we know that there's something potentially inhibitory in that sample, and we'd have to do dilution and reanalysis of that sample. But that way we can kind of have a high degree of confidence in every single sample, make sure that we know the number we're getting is a good number, and we have confidence in that result. So you can see that for a 96-well plate, we're not getting a whole lot of samples on there. And that's kind of by design, because we are trying to make sure we have enough QC to have confidence in this procedure. And also we run this in duplicate. So this would be what the plate would look like for N1, for example, and then we would run another one exactly the same for the N2 probe primer set, because these are simplex, they're not multiplexed assays. So amplification plots look kind of like you'd expect. This is the N1 amplification plot for calibration curve. Our 5 points are there, and it looks pretty much what you expect. It's reasonably sensitive at the low level, and we did find that the N1 probe primer set is more sensitive than N2, just by a little bit. We calibrate kind of the same, but it's a little bit more sensitive. Calibration curves also look good, very linear, good response, good reproducibility. So we're pretty happy with how that turns out. And when we're doing this concentration, we're actually doing a 50 ml sample to a final extract concentration of 100 microliters. So we have a 500x concentration factor. And because we're using 2.5 microliters per reaction, we get a detection limit of about 8,000 copies per liter of raw wastewater. Now, again, the N1 is a little more sensitive than N2. We find that this is a kind of a reasonable number to work with, and it seems to capture most of what we need for this method, at least. So when we were developing this procedure, we kind of wanted to make sure that it would work for most everything. The issue here is that wastewater is complicated. It's ranges from really, really clean to pretty nasty. I mean, you have the whole gamut here, and it could be due to industrial discharge or just residential discharge or agricultural or stormwater or whatever. So we wanted to make sure that we'd have a method that would work for every kind of sample we would receive, or a vast majority of the samples we receive. So we tested out a bunch of different extraction techniques, concentration extraction steps. These are some of the ones we tested. So we tested precipitation, the crashing out the virus with a peg and spinning it down. We tested filtering it, then crashing it out and then spinning it down. We got kind of low recoveries for some of the matrices we tested with this, so it wasn't so robust for our types of samples. Then we settled on using this molecular weight cutoff filter at 10,000 Daltons, which worked pretty well. It was easy to use. It captured most of the virus from a lot of different types of matrices. And we first tested it without any pre-filter, so we just would put sample right in there and see what happened. And as you can imagine, for clean extracts or clean wastewater samples, not a big deal. For dirty stuff with a lot of junk, it would foul up those filters pretty quickly, and it really didn't work out very well to pass 50 mils of sample through one of these little filters. So we ended up settling on that filtration up front. So we did 0.22 micron filtration to get rid of the solids, and then follow that way at concentration with the 10,000 Dalton molecular weight cutoff filters. So after we have that extract, it's still a bit dirty. I mean, that extract still has a lot of dissolved stuff in it. Like I mentioned earlier, we have organics, metals, all kinds of things that are potentially inhibitory. So we wanted to figure out a way to get rid of that bad stuff, the stuff that would mess up the reaction and give us some bad data on the experiment. So we tested out a couple different approaches for RNA extraction. First one we did was just a stabilizing buffer. Figured it'd be the easiest. As you can imagine, for clean samples it worked. It's like the start of everything. Dirty samples, it did not work very well. So anything that was really contaminated, really dirty, all that stuff would stay in the sample and have a lot of inhibitory effects when we did PCR in it. So we stuck with an extraction technique like magnetic beads or spin columns. We tested both out, and we found that the magnetic beads worked pretty well, but it was a bit more cumbersome. Sometimes the extracts would be a little too dirty, and we'd get some weird effects in the magnetic bead separate or pulling the beads out and resolubilizing them. So it was a bit more cumbersome for us. But the spin column was great. The spin column was nice because it has a pre-filter. We were able to really remove a lot of the junk. We have a couple of extra washes in there so that we can make sure that everything comes off and we have a relatively clean extract. So we settled on that, and it gave us pretty good recoveries and pretty good efficiencies for most of our samples. So when we're testing this out, developing it, we're getting greater than 50% recovery on matrix spikes and greater than 65% for the blank spikes. So we were pretty happy with how this procedure worked out. And it was nice because it's pretty easy to deploy in our lab. So we wanted to make sure we had something that would be straightforward and it had good recoveries and easy to deploy and straightforward to use. So it wasn't so complicated and so cumbersome and time consuming in the lab. So here's a little bit of an example of how it would look in a real world sample. So this is for a residential community where they had minimal people living at the spot up front. So you have a baseline, right, for a couple of days of low infectivity. And as soon as people started moving in, you get a sharp increase in the rate. So you can see the trend was increasing, right? More people were there. Makes sense. And the idea here would be that you would continue to monitor this every day, every couple of days, to get an idea of whether that trend continues to increase or it stabilizes and starts decreasing. So in this case, you can see it already starts to stabilize, and hopefully it'll start decreasing, showing that the infection rate in that community, in that population, is going down. So this is how you would kind of use this data over the course of a period of time. And it has to be, like I mentioned earlier, frequent testing to make sure you have as many time points as possible to get that resolution. Because this data wouldn't be very useful if you just had day 1 and day 12. It wouldn't tell you a whole lot. But if you have all these time points in between, you get an idea of what's actually going on. So to sum up the whole approach to wastewater-based epidemiology is pretty useful. It's very powerful. It gives a lot of useful information to folks who are making decisions. Kind of adds another data point in their equation of what to do with society, with opening, closing, predicting outbreaks and things like that. So I think it's very useful in conjunction with things like contact tracing and individual testing, because it does serve as a useful indicator. It also gives that advantage of being a little bit quicker than individual testing, so you can have kind of a jump on something that's maybe going to happen. Say, a lot of universities now are doing this kind of testing in their dorms so they can see when an outbreak is about to start or happen or come up before people actually start showing up sick at their health center. So some of the protocol here, we take a 50 ml sample, pasteurize it for 90 minutes at 75 degrees. And the idea here is that we want to eliminate all the nasties, not just the SARS-CoV-2, but all the E. coli and the potentially pathogenic other agents in the sample. We filter it through a 0.22 micron filter to remove a bulk of the total suspended solids in the sample, make it a little bit easier to do the concentration, which we do in a 10,000 Dalton molecular weight cutoff filter. Then we pass the whole 50 ml volume to that. We concentrate it down, do the RNA extraction to remove the remaining matrix components, things like organics and inorganics that could potentially interfere with the PCR, and we use that absolute total RNA for this. We wind up with a 100 microliter extract, and we do the analysis with qRT-PCR. Pretty good results, pretty good matrix spike recoveries, blank spike recoveries, and good sensitivity on the system itself. So with that, I'll pass it back to Christy, and I'll answer some questions at the end of the talk.
Great, thank you, Dr. Pieri. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'll conduct the Q&A portion of the webinar after the presentations have concluded. Next up is Dr. Israeli. Please go ahead.
Thank you very much. And now I'm going to be talking about applications of qRT-PCR workflows for research in SARS-CoV-2. So specifically on today's agenda, I would like to talk about the workflow and the key benefits of the reagents and kits which we offer, as well as show some data examples and also go through the portfolio of qPCR reagents. So as we've heard from Augustin, pandemic and epidemic events are happening at greater frequencies over the past few years. And as this is happening, and as a way to meet the challenge of those ever-increasing events, the more methodologies are being developed to really detect these pathogens in the environment and specifically in wastewater. And having the right bioanalytical tools to do this is very important. So at Agilent, we offer a number of bioanalytical tools in order to do this. And so we really serve scientists in a number of industries, including biopharma, infectious disease, clinical diagnostics, and life science research. And we offer many different platforms to do this. Now, you may be familiar with Agilent's presence in water and wastewater analysis regarding our instrumentation. And we have a number of instruments and a long history of this presence. And so you may be familiar with our gas chromatography, liquid chromatography, and mass spectrometry instruments that have been used in the analysis of contaminants in water and wastewater. And more recently, we've been forming collaborations with groups in order to develop new methods to really look at use of potable reuse treatment of water and those sort of things. So our instruments are out there in water and wastewater analysis. And while that's the case, we have also other types of solutions and tools for labs in analysis and research. And within infectious disease, there are other types of genomic methods, as Augustin covered, to really get at detection of infectious diseases and pathogens in the environment and wastewater. And so today I'm really going to be focusing on quantitative PCR and PCR analysis. And today, really, there's widely used qPCR and PCR in the detection of SARS-CoV-2 in clinical diagnostics. And there are a number of reasons for this. So real-time PCR really offers quite a few things in this regard. So it offers a fast workflow for a large number of samples. And it's also a cost-effective solution in the detection of infectious disease agents. So not only RNA-based, but also DNA-based. And so what I would like to do is take you through the qRT-PCR assay workflow. So again, this is quantitative reverse transcription polymerase chain reaction. And what I'd like to do is discuss those products which we have, which address those different parts of the workflow. So for RNA isolation of viral RNA, we have the Absolutely RNA Isolation Kit, which is available in different formats. And once the viral RNA is isolated from the sample, or say from a water sample, our Brilliant 3 probe qRT-PCR master mix can be used to convert the RNA to cDNA with a reverse transcriptase, and then amplify those specific viral targets with DNA polymerase. And so in order to detect that amplification, our ARIA-MEX real-time PCR instrument, in conjunction with consumables and the software that comes with the instrument, can be used for data analysis. So really, we have many of the components that are required from sample to data analysis here. And what I'm going to do is really just highlight a couple of these and their benefits in the workflow. So starting with the Agilent Absolutely RNA Isolation Kits, they yield high amounts of RNA. It's a short protocol. It requires only 20 to 30 minutes per sample. And it accommodates really a variety of sample volumes and formats. It can be used with complex matrices such as wastewater, concentrated wastewater. And there's a high sensitivity of the kit with the limit of detection of about 10 copies per sample. And it can take volumes of 100 to 600 microliters, depending on the kit that's being used. As Augustin mentioned, this is a filter column-based method, so it can be used either manually or in an automated method. And if used manually, you only require centrifugation or a vacuum manifold. And the recovery for the Absolutely RNA kits are quite high at 90% recovery. So once the RNA is isolated, it can then be taken through the process further on with our Brilliant probe master mixes. And we offer different kinds of Brilliant probe master mixes. They all employ a novel fast activating chemical hot start technology. And what that does is it enhances amplification specificity and speed, and that's important for target detection, specifically for the viral targets. And what I also want to highlight here is that these Brilliant master mixes really have a great deal of multiplexing capabilities. So anywhere between 2 to 4 reactions, if not more, per sample type. So in other words, per tube. And that's very useful if one wants to multiplex different viral RNA targets, such as N1, N2, and also any other kinds of controls, sort of fecal matter controls, that sort of thing. There's a high resistance to inhibitors, which improves analysis results for challenging and degraded samples. And there's also good reproducibility. And so there are 2 versions of this kit. There's our Brilliant 3 qRT-PCR kit. And I do just want to remind the audience that the RT here stands for reverse transcriptase. And we also have a Brilliant 3 and a Brilliant 2 kit. And they've both been validated on fast and standard cyclers, respectively. Their sensitivity is high at 5 to 10 copies per PCR reaction. And, you know, we understand that when users are trying to develop an assay, they often want the flexibility of being able to optimize with standalone reagents. And so our Brilliant 2 kit is offered not only as a one-step format, but also as a kit with standalone reagents, which would be able to allow you to do that. Our Brilliant 3 kit is offered as a one-step solution. Again, these kits are open platform kits. They can be used on our ARIA-MEX real-time PCR system, as well as comparable real-time PCR cyclers. Now, this particular brilliant qRT-PCR kit really takes you from RNA to amplified DNA. But as in the case where one may want to optimize the cDNA synthesis or the reverse transcriptase portion of the workflow, we also offer a separate reverse transcriptase, and that is the affinity script. And then in that case, if the affinity script is used, a brilliant qPCR kit can be used to amplify DNA after that step. So our cDNA synthesis kit yields high amounts of DNA across a broad range of temperatures. And that's important because at high temperatures, it enables the transcriptase to read through RNA with high secondary structure. And as well, the long enzyme half-life at 55 degrees enables transcription through long fragments, specifically GC-rich fragments, which can also contribute to high secondary structure within the RNA. And so the sort of target lengths that one may anticipate getting are 20 kPs, if that's the target length that you're looking for. The sensitivity of the reverse transcriptase is very high, so as low as 3 picograms per sample. So that's 3 picograms of RNA. There's high activity in the presence of inhibitors, and again, high yield for GC-rich regions. Reaction time for these kits is 15 to 25 minutes. Now, Augustin really spoke very nicely about the use of the reagents in the workflow in the detection of SARS-CoV-2 in wastewater analysis. And if we remember previously that this is really sort of a continuum. So being able to detect the SARS-CoV-2 in wastewater has implications in diagnostics as well. And so we're actually really pleased that we've been able to find that our SARS, we and our partners have been able to find that our reagents work well in SARS-CoV-2 detection in all kinds of samples and all kinds of applications, including samples from individuals, again, for detection of SARS-CoV-2 there. And so this slide is showing that work that was done in partnership with Sinai. They're a group in Brazil, and they were looking at the presence of the virus in samples, specifically nasopharyngeal samples. And so here, they essentially have been able to use the similar products for a slightly different application. And so in their protocol, they use the absolute RNA isolation kit. And here what they did actually was very interesting. They were able to optimize an automation protocol for the RNA isolation kit using the Agilent Bravo liquid handling platform. And once they had their RNA, they used the qRT-PCR Brilliant 3 master mix kit to convert the RNA to cDNA and then amplify the DNA. And again, use the RAMX real-time PCR system in that method. And so again, we're able to see that the limit of detection is quite good with high sensitivity, up from 200,000 RNA copies per ml down to 2 copies per ml. And so so these can be used across a broad range of methods and applications, these reagents. So we also offer other reagents which can be used in SARS-CoV-2 detection. Augustin had mentioned a lysis and stabilization buffer. That's our sidestep, and that is for extraction of RNA. We have other types of kits for also DNA isolation for pathogens, which are DNA-based, and those are on our right-hand side, the RNA extraction kits, as well as specialty master mixes and controls and references, which one might want to use to detect any types of inhibitors that may be inhibiting their qRT-PCR reactions. And so with that, I do want to say thank you. And for more information about us and the types of solutions that we offer, please visit us at www.agilent.com. And thank you for your attention. And I think now we'll take some questions.
Great. Thank you very much, Dr. Israeli. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And before we begin the Q&A, we'd like to ask attendees to take a moment after the webinar has ended to give us feedback by taking our exit survey. All right, so let's start the Q&A. Augustin, our first question is for you. Have you been able to isolate any viable SARS-CoV-2 virus using your method?
So that's a good question. We actually purposefully do not keep anything alive. So we pasteurize the samples pretty much as soon as they get in. So we are not able to get anything viable out of that. I have seen in literature that some folks have been able to recover some viable SARS-CoV-2 in wastewater, but with our procedure and how we do things in our lab, we don't have that option. And it's also because we're a little more risk averse, so we don't want to take any chances with anything potentially infectious.
And we have a question about, is there a basis beyond intuition? Has there been any research that's been published for relating the RNA concentration in PCR-amplified samples to infection level in the population?
So that's a good question. We kind of get that question often from our customers. And that's something that there's a lot of modeling out there, and you can model stuff and take certain assumptions into consideration and get an idea of what's there. But it's very difficult to get a good, accurate number with the degree of confidence I would want from an analytical measurement, because it just has a lot of factors. It depends a lot on, what the wastewater stream is made out of, what is the source. Like if you have somebody who's early in the infection, late in the infection, they may be shedding different amounts of virus. What is the amount of fecal matter in the wastewater stream per person? So it's just a lot of assumptions you need to make to do that. And I know that some folks are modeling it, but it's something that we're just not comfortable giving a number, because of those assumptions that need to be made.
Can you explain to us a little bit more about how you validated your assay?
Yeah, so the validation was done through a matrix spikes. We positive spiked matrix samples and did replicates and see what worked with different extraction procedures. And once we settled on one that we liked that gave us good recoveries, we did matrix spikes at different concentrations in different types of matrices to validate that.
Can you talk to us a little bit about what method of extraction you use or that you recommend, and what volume of input wastewater you use for the extraction? For example, is 50 milliliters of sample enough, or do you recommend more or less? And Sharon, if you would like to chime in on that as well, that would be great.
Yeah, from my end, I think always the more sample you can use, the better, right? So if you have a larger sample volume, say you have 10 liters, you'd have a better idea, a more representative sample of what you're working with, and potentially lower limits of detection. Now, if it's not obvious to me what the absolute best limit of detection is, like, are you looking for one copy per liter? Maybe that might be a little bit of overkill. But yeah, the more sample you use, the more sensitive you'll be, and the more representative of a sample you'll have. The issue becomes using like a few liters of sample of clean water is pretty straightforward, but a few liters of a raw sewage wastewater influent is going to be pretty tough to clean up, and it would require a lot more effort than it would be reasonable, I think, to get this number. So we settled on 50 because it was kind of an easy number where we got a reasonable concentration factor and didn't have a huge contribution from the matrix, something we could easily mitigate with the RNA cleanup.
Sharon, do you have any thoughts?
Well, yeah, so I can say for the absolutely RNA isolation kits, right, so they can take between 100 to 600 microliters of sample, depending on the version of the kit that's used. And what you're really getting is extracted and isolated RNA. Our side step is just extracted, so there may be some more issues with inhibition and qRT-PCR, but both may be used.
Augustin, could you tell us a bit about your, was your approach inspired by monitoring sewage for the polio virus? And if there's any similarities or differences between your approach and that approach?
Yeah, it totally was. Like we looked at the literature and what other people have done historically with wastewater-based epidemiology, and things like polio or norovirus have been done a lot in the past, and we based our approach kind of on that and tried to essentially leverage the best technology we had available now to get the best data we could in our lab with the the ease of use that we kind of wanted. So yeah, it was definitely based on things like looking at polio in wastewater and other historical uses of wastewater-based epidemiology.
And can you tell us if you've had any requests to test for a panel of pathogens in addition to SARS-CoV-2? Do you think it's viable, for example, to carry out shotgun sequencing of wastewater samples in order to observe co-occurrence of pathogens that might affect the characteristics of the outbreak?
Yeah, that's actually a great question. That's something that we would love to do. We've had discussions a little bit about that, and that's like the goal, right? To be able to do this in an ongoing fashion. I think it would be huge from a public health standpoint to be able to do that shotgun sequencing and see what's going on in wastewater in a population. Like, for example, if you could predict a future outbreak or potential future outbreak of a particular disease or illness would be very helpful. So that's something that we are interested in pursuing. At the moment, there's not a lot of demand for that. I think it's kind of a niche request, but it's something that we're definitely looking at and would like to get going sometime soon.
With combined sewage systems, do you correct for rainfall? Is there a method for that?
So that's something that would have to be taken into account. I mentioned that also, like if you do have a significant contribution from stormwater in your treatment plant, you'd have to correct for that. So you'd have to look at the flow proportioning on your compositing device and take into account the contribution from wastewater. And that's not something that we do. We could, I guess, but typically what we'll give is a number to our customers that will have, a trending indicator from that, and then have them kind of work out the math from the contribution. But that definitely needs to be considered when you're looking at this data.
And then as a follow-up to that, is there a point at which, when you're collecting composite samples, that you'd be concerned about getting false negative results? And then how do you account or correct for that?
Yeah, so the false negative would come if you're, a lot of times that would come with like a grab sample, not so much a composite if you're maybe pulling a sample when there's very little contribution. But a false negative would pretty much only be due to maybe a detection below the reporting limit, in which case it would be a true negative based on our detection limit. A false negative where you have the extra or the virus in the sample would be pretty difficult if it was in appreciable concentrations, but it would, of course, require careful planning on your collection and your compositing device.
You may have mentioned this, but if you could tell us again, does your method detect live virus or does it detect residual RNA?
So it only looks at residual RNA. So we aren't, we're not really concerned with looking at the live virus. I know there are some folks that are doing that. But the EPA is also looking at trying to see what fraction of live infectious viruses left in the wastewater. But in our case, we're pasteurizing everything. So we're just looking for the residual RNA and non-infectious stuff.
Any kind of limit of detection that would give an estimate of which proportion of individuals have to be infected within a given group where you'd be able to detect SARS-CoV-2 in wastewater, or, for example, a method that would determine the number of infected individuals in a catchment based on the concentration of SARS-CoV-2 in the wastewater?
Yeah, so it's kind of the same thing, that it's probably you can, but it would require a significant amount of modeling and a lot of assumptions to be made to get that kind of correlation. And I, from my experience, I wouldn't recommend it because of the just assumptions that need to be made to kind of correlate that.
So we've had a couple of questions about the matrix spikes. What level of recovery are you achieving with your matrix spikes? You mentioned that you had 175% at one point. Is that an outlier or is that more of an average?
So that was the range that we observed. Some of these would have been, so we're spiking real matrix. So if it had some incurred virus in the sample, we tended to see a little bit of a high bias on those. Generally speaking, the average would be lower, close to the 50 to 70% recovery, but those were the extremes of what we saw, the 50 to 175. And the 175 was an incurred sample that had some in there as well.
And what is the matrix spike being spiked into? Is it clean water or wastewater? And is that done for every sample tested to assess each specific recovery rate? And then to follow up on that, how do you explain the greater than 100% recoveries?
So the matrix spikes are spiked in matrix, so not in clean water. These are going to be in a real sample. So let's say we get 20 samples in a batch. One of those samples we will run with the matrix spike and without the matrix spike. And the high recovery I just mentioned is likely due to a sample that already had an incurred amount of SARS-CoV-2 in the sample. So we got a little bit of a high bias there because of the spike level being much lower than the incurred value. But the spiking is done always in a real world matrix of a sample that's being analyzed at the same time. So when we did the development, we did look at blank spikes where we were spiking clean water and looking at absolute recovery, but that's not really representative of the samples we're running. So when we run batch work, we're just doing that matrix spike to assess the whole procedure.
Can you talk to us a little bit about what the mechanism is for a respiratory virus to appear in fecal matter, and how long the virus is active or infectious once it gets to the GI system and then discharged into the waste system?
So, I'll admit I'm a chemist, so I'm not so well versed on the what happens to the body when you have an infection. So I can't answer that question. But from the literature I've seen, it appears that the fecal the presence in the GI tract happens kind of right away, usually before the onset of respiratory symptoms, and it can linger for much longer than your respiratory symptoms. So it kind of gives a worst case scenario measure, right? If you have somebody who has respiratory symptoms that start on day 8 of infection and end on day 12, you could potentially have viral shed going through the GI tract from day 2 to say day 20 to have that kind of coverage. So it's something that is more I guess prevalent or it has a longer lifetime than the respiratory symptoms, but I don't know how it actually gets into the GI tract, unfortunately.
Could you talk to us a little bit about your sample collection methods? Do you use automated methods, or do you prefer other types of sample collection?
Yeah, so for sample collection, I prefer automated methods. Like I mentioned, the move is to do a composite over, like, say, a 24-hour period where you have a device, a sampler that would collect alcohol as a sample throughout the course of 24 hours and get one composite sample. Now, if you do it manually, it's doable, but it's very cumbersome to be there and doing that for a whole day. So there are a lot of these systems that exist to be able to facilitate that. But I definitely recommend doing an automated 24-hour composite versus a single grab manual sample.
So we've had a question about running a higher number of sample replicates. I believe you said you used 2, but would it be possible to run a higher number of replicates in order to decrease the limit of detection?
So we're running replicates more for reproducibility to see the RSC, make sure that the numbers we're seeing are correct. Not so much to lower the limit of detection. Now, we could change the amounts of sample that we're using. We could run more replicates and try to push that limit of detection a little bit lower. But there hasn't seemed to been a need so far from our customers to push it lower. But as we do get requests and people start looking at trying to push it lower, we would probably change that to kind of get a little bit lower LODs. But for now, it seems to be suitable for what people are looking for.
Okay. If you're doing a monitoring program, let's say on a college campus, and you have a low positive rate starting out, how frequently do you think you would collect samples in order to do ongoing monitoring?
I would definitely look at every couple of days. I think if you have a low rate at first, the risk is there for maybe a spike to happen. Say there's one infected person comes back to campus and, I don't know, has a party or mingles or whatever, and then you could have a quick increase there. But I would definitely say, every couple of days would be a reasonable kind of time window. The more frequent, the better. Obviously, that's more resolution on your measurements. But then it becomes a little bit more complex and more expensive and more cumbersome to do sampling every day. So I would recommend every couple of days probably.
And were there any internal controls that you amplified in addition to the N1 and N2 genes?
So there aren't. That's why we're doing the inhibition control separately, and we do that with the RNA, the extracted RNA for SARS-CoV-2. We're working on adding an internal control as a fecal indicator. So that's something that's coming down the pipe that we're going to be hopefully rolling out here in a little bit for our method. But as of now, as the method is today, we don't have that. We have to run it in a separate well.
All right. I'm afraid we've run out of time for today, so we're going to wrap it up there. We'd like to thank Augustin Pieri, Sharon Israeli, and our sponsor, Agilent Technologies. If we didn't have time to get to your question, we will follow up with our experts. As a reminder, please look out for the pop-up survey after you log out to provide your feedback. If you missed any part of this webinar or wish to listen to it again, a link to an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.